What term was originally used for DNA?
DNA was originally called nuclein because it was first isolated from the nuclei of cells. In the 1860s, Friedrich Miescher, 1844–1895. A Swiss biochemist working in Germany at the University of Tübingen lab of Felix Hoppe-Seyler, 1825–1895. Was given the task of researching the composition of white blood cells. He found a good source of white blood cells from the used bandages that he obtained at a nearby hospital. He washed off the pus and isolated a new molecule from the cell nucleus. White blood cells have very large nuclei. He called the substance nuclein, which we now call DNA. The substance was rich in nitrogen and phosphorus and also contained carbon, hydrogen, and oxygen. Hoppe-Seyler checked and verified the important work of his student Miescher.
Why were such dangerous chemicals as DDT, PCBs, and CFCs released into the environment?
Dichlorodiphenyl trichloroethene, DDT, polychlorinated biphenyls. PCBs, and chlorofluorocarbons, CFCs, were once widely used. Although DDT was synthesized as early as 1874 by Othmar Zeidler, it was the Swiss chemist Paul Müller, 1899–1965, who recognized its insecticidal properties in 1939. He was awarded the 1948 Nobel Prize in Physiology or Medicine for his development of DDT. Unlike the arsenic-based compounds then in use, DDT was effective in killing insects and seemed not to harm plants and animals. In the following 20 years it proved to be effective in controlling disease-carrying insects. Mosquitoes that carry malaria and yellow fever, and lice that carry typhus, and in killing many plant crop destroyers. Publication of Rachel Carson's book Silent Spring in 1962 alerted scientists to the detrimental effects of DDT. Increasingly DDT-resistant insect species and the accumulative hazardous effects of DDT on plant and animal life cycles led to its disuse in many countries during the 1970s. In fact, DDT and PCBs have been added to the list of chemicals known as estrogenic compounds, that is, synthetic substances in the environment that cause the mammalian body to respond as if to estrogen. A group of chemicals with the same general chemical structure and physical properties as DDT are known as the polychlorinated biphenyls or PCBs. Because of their physical properties, non-flammability, chemical stability, high boiling point, and electrical insulating properties, PCBs can be used in a variety of applications. Formerly, many products contained these compounds. From electrical circuitry to the dyes and pigments used in paint, to carbonless copy paper, all were manufactured with PCBs. Before production was ceased in 1977, the United States produced about 1.5 billion pounds. 6.8 billion kilograms of PCBs. Chlorofluorocarbons, CFCs, are commonly used as aerosol sprays. Refrigerants, solvents, and foam-blowing agents. They are in and of themselves non-toxic and non-flammable molecules containing chlorine, fluorine, and carbon. However, they are thought to have a deleterious effect on ozone concentrations in the atmosphere.
What is a reflex?
A reflex is an involuntary response formulated in the spinal cord to a specific stimulus.
What is the difference between white rot fungi and brown rot fungi?
White rot fungi, found in the wood of deciduous trees, first attack the lignin of wood. Once the lignin is digested, the fungi destroy cellulose and other major parts of cells. The partially decayed wood with residual cellulose is off-white in color. Hence the name white rot fungi. Brown rot fungi, found in conifers. Damage the cellulose first but do very little, if any, damage to the lignin. The name brown rot fungi came about because infected wood becomes dark reddish-brown to golden in color.
What technique may be used to determine the presence and concentration of various chemicals in a solution?
Spectrophotometry is based on the principle that every different atom, molecule, or chemical bond absorbs a unique pattern of wavelengths of light. Using a spectrophotometer, scientists are able to measure the amount of light that is absorbed or transmitted by molecules in solution. This value of this measurement allows scientists to identify the chemical.
What are the largest human genetic databases?
Genomics research on entire populations is an area that is rapidly expanding. In order to control access to genetic information and provide ethical guidelines for its use, several countries have developed human genetic databases. Iceland and the United Kingdom have national genetic databases, while Canada and Sweden have regional databases.
What are the steps to prepare a specimen for examination?
The three basic steps to prepare a specimen are fixation, preservation, staining, and mounting. Preservation prevents destruction or decay of the specimen as well as inhibiting microbiological growth. Different stains and dyes attach to different parts of a cell, such as the nucleus. Specimens are mounted in a medium that is also a preservative and often covered with a cover slip.
Are humans still evolving?
In order for humans to continue evolving in a measurable way, we would predict that those individuals with certain variations alleles of traits must reproduce more efficiently than those without those alleles, such that eventually the alleles in question become more common in the population. For example, if nearsighted individuals were less likely to produce healthy offspring, for example, perhaps they have trouble finding enough food, then we would expect that eventually there would be very few nearsighted individuals in the population, assuming that nearsightedness is genetic. Because of our uniquely human skill of inventing things like eyeglasses and corrective surgery, which counteract genetic programming without actually changing the genes, there is little selective pressure against being nearsighted, so it is unlikely to be lost from the human population gene pool. The same could be said for many other traits. So while our gene pool could certainly change with time, it is unclear whether it will change in a clearly measurable direction such that we physically evolve.
What is a stem cell?
Stem cells are undifferentiated cells, meaning that they do not have a specific function that are capable, under certain conditions, of producing cells that can become a specific type of tissue. Stem cells present in adult humans are found in bone marrow and other tissues, such as fat. However, most research interest is focused on stem cells present in fetal tissue, which, in a laboratory setting, can reproductively divide indefinitely and be stimulated into becoming a variety of different cell types. The potential benefits of stem cells have made the research of these cells an exciting research topic.
Which antibacterial products are the most effective for general use?
Non-residue producing agents, such as hydrogen peroxide and bleach, are effective agents for controlling microbes. Several consumer products with residue-producing agents have proved to be effective for specific conditions. For example, antibacterial toothpaste helps control periodontal disease. Antibacterial deodorants suppress odor-causing bacteria, and anti-dandruff shampoos help control dandruff.
Is alcoholism an inherited disease?
If all of the adults in the U.S. were to be tested, an estimated 10% of the adult population would be classified as alcoholic. Of that number, the ratio of males to females is approximately 4:1. This disproportion could be attributed to both environment and genetics. Research involving male relatives, both biological and adoptive, points to a multifactorial source for alcoholism. In other words, while sons and brothers of male alcoholics are likely to be alcoholic as well. 25% and 50%, respectively. Genetic inheritance is not solely responsible for all of the cases of alcoholism. Alcoholism has been linked to depression, as more is learned about the genetic basis of mood disorders. The genetic basis of alcoholism may be unraveled.
What is a gene probe?
A gene probe is a specific segment of single-strand DNA that is complementary to a desired gene. For example, if the gene of interest contains the sequence OTKaka, then the probe will contain the complementary sequence TACT. When added to the appropriate solution, the probe will match and then bind to the gene of interest. To facilitate locating the probe, scientists usually label it with a radioisotope or a fluorescent dye so that it can be visualized and identified.
What is a fiddlehead?
The type of fern typically grown as a houseplant is of the diploid, or sporophyte, generation. It is composed of a rhizome, an underground stem that occurs horizontally, which produces roots and leaves called fronds. As each young frond first emerges from the ground, it is tightly coiled and resembles the top of a violin, hence the name fiddlehead.
What are autosomes?
Autosomes are chromosomes that contain information, gene sequences, available to both sexes. These are the chromosomes commonly used in genetic examples.
What is an ecological pyramid?
If the organisms in a food chain are arranged according to trophic levels, they form a pyramid, with a broad base representing the primary producers, and usually only a few individuals in the highest part of the pyramid. Also known as a pyramid of numbers, an ecological pyramid is a way of describing the distribution of energy, biomass, or individuals among the different levels of ecosystem structure.
Which group of animals has exceptionally well-developed brown fat?
Mammalian hibernators have exceptionally well-developed brown fat. Some scientists even refer to it as the hibernation gland. The supply of brown fat is used during the winter months when the animal hibernates and is built up during the spring and summer. Brown fat is important in the revarming process that the animal undergoes after hibernation.
What is meant by genetic determinism?
Genetic determinism is a theory proposing that behavior and character are entirely shaped by genes. Since only genetic material passes from one generation to the next, it seemed logical at one time to assume that all of our traits, physical, behavioral, psychological, were the result of gene sequences. More recent and extensive research has demonstrated the importance of environmental factors in determining how or which genes are actually involved in the development of the individual.
What are the characteristics of the protists?
Protists are a diverse group of organisms. All protists are eukaryotic. Many are unicellular, but they may be multicellular, multinucleate, or exhibit a colonial organization. Although most are microscopic, some are much larger, reaching lengths of nearly 200 feet 60 meters. In early, traditional taxonomic schemes, they were united on the basis of being neither plant nor animal nor fungus. Current evidence suggests that protists exhibit characteristics of the plant, animal, and fungi kingdoms.
How much of the human genome actually codes for genetic information?
Only about 5% of the human genome appears to code for genetic information.
How do animals know which direction to travel?
The ability to navigate from one place to another is found among a diverse group of species. While it is well known that birds can navigate over long distances, many people are unaware that bats, salmon, locusts, and frogs are also capable of this behavior. Animals use a variety of cues to find their way, including the position of the sun, moon, and stars. Topographic features of the landscape, meteorological cues, e.g., prevailing winds and magnetic fields are also used by certain animals.
Can we make artificial cells?
Research in progress at the National Aeronautical and Space Administration, NASA, is focused on artificial cells as a means to deliver medicine in outer space. These cells are able to withstand dehydration and thus can be safely stored for long periods. Artificial cells are made of a polymer that acts like a cell membrane. But the polymer is stronger and more manageable than real membranes. These polymers are called polymersomes and can be made to cross-link with other polymers. Researchers feel that many different kinds of molecules can be encapsulated within these polymersomes and then delivered to specific target organs. An example would be an artificial blood cell that not only delivers oxygen but also medication as it travels through the body.
What is thermodynamics?
Thermodynamics is the study of the relationships between energy and the activity of a cell. The laws of thermodynamics govern the way in which cells transform chemical compounds. The first law of thermodynamics states that the total energy of a system and its surroundings will always remain constant. The second law of thermodynamics states that systems, such as a cell, tend to become disordered. Disorder usually arises in the form of heat. Entropy is the term that refers to the disorder that tends to disrupt cells, or even the universe.
How is petrified wood formed?
Petrified wood is formed when water containing dissolved minerals, such as calcium carbonate, CaCO3, and silicate, infiltrate wood or plants. The foreign materials either replace or enclose the organism, so the structural details of the plant are retained. The process takes thousands of years. Botanists find these types of fossils to be very important since they allow for the study of the internal structure of extinct plants. After time passes, the plant or wood seems to have turned to stone because the original form and structure are retained, but it does not actually turn into stone.
What types of samples can be used for DNA fingerprinting?
Any body fluid or tissue that contains DNA can be used for DNA fingerprinting, including hair follicles, skin, earwax, bone, urine, feces, semen, or blood. In criminal cases, DNA evidence may also be gathered from dandruff, from saliva on cigarette butts, chewing gum, or envelopes, and from skin cells on eyeglasses. What is the largest forensic DNA investigation in U.S.?
What is the Kelvin temperature scale?
Temperature is the level of heat in a gas, liquid, or solid. The freezing and boiling points of water are used as standard reference levels in both the metric centigrade, and the English system, Fahrenheit. In the metric system, the difference between freezing and boiling is divided into 100 equal intervals called degree centigrade, degree C. In the English system, the intervals are divided into 180 units, with one unit called degree Fahrenheit, degree F. But temperature can be measured from absolute zero, no heat, no motion. This principle defines thermodynamic temperature and establishes a method to measure it upward. This scale of temperature is called the Kelvin temperature scale, after its inventor, William Thomson, Lord Kelvin. 1824 to 1907, who devised it in 1848. The Kelvin, symbol K, has the same magnitude as the degree centigrade. The difference between freezing and boiling water is 100 degrees, but the two temperatures differ by 273.15 degrees. Absolute zero, which is minus 273.15 degrees Celsius on the centigrade scale.
What does genetics have to do with behavior?
Some ethologists feel that all behavior is genetically programmed. If a behavior is under genetic control, then it is a sequence of events. Sign stimulus, releasing mechanism, and fixed action pattern. However, the mainstream view is that most or all behavior is a result of a combination of genetic programming and environmental learning.
How do animals communicate by scent?
A variety of animals use scent marking to identify their territory. Some use scent marking for possessions. In Asian muntjac deer, males have scent glands on their faces that they use to mark their mates.
Who was the first person to study populations mathematically?
Thomas Robert Malthus, 1766 to 1834, in 1798 attempted to inform people that the human population, like any other population, had the potential to increase exponentially. Malthusian ideas were not well received, as he predicted the rate of population growth would exceed the ability of the land to produce food. His work was later used by Charles Darwin to explain his theory of natural selection.
What are the main functions of the plasma membrane?
The main purpose of the plasma membrane is to provide a barrier that keeps cellular components inside the cell while simultaneously keeping unwanted substances from entering the cell. The membrane allows essential nutrients to be transported into the cell and aids in the removal of waste products from the cell. The specific functions of a membrane depend on the kinds of phospholipids and proteins present in the plasma membrane.
What are diatoms?
Diatoms are microscopic algae of the phylum Chrysophyta in the kingdom Protista. Almost all diatoms are single-celled algae and dwell in both fresh and salt water. They are abundant in the cold waters of the northern Pacific Ocean and the Antarctic. Diatoms are yellow or brown in color and are an important food source for marine plankton and many small animals. Diatoms have hard cell walls. These shells are made from silica that has been extracted from the water. It is unclear how the extraction of silica from water is accomplished. When they die, their glassy shells called frustules sink to the bottom of the sea and harden into rock called diatomite. One of the most famous and accessible diatomites is the Monterey Formation along the coast of Central and Southern California.
Which organism is known as the humongous fungus?
The humongous fungus is an enormous, underground fungus that grows in northern Michigan. The fungus, Armillaria gallica, was discovered in 1992. It encompasses 35 acres, 15 hectares, and is thought to be at least 1,500 years old. Scientists proved that the organism was a single fungus by taking 20 samples of the fungus and performing DNA analyses on 16 fragments from each sample. The genetic material of each sample was identical.
What are the two types of euryhaline fishes?
There are two types of euryhaline, from the Greek terms euries, meaning broad, and hals, meaning salt, fishes. One type includes flounders, sculpins, and killifish that live in estuaries or intertidal areas where the salinity of the water fluctuates throughout the day. The second type includes salmon, shad, and eels that spend part of their life cycles in fresh water and the balance in seawater.
How did the dodo become extinct?
The dodo became extinct around 1800. Thousands were slaughtered for meat, but pigs and monkeys, which destroyed dodo eggs, were probably most responsible for the dodo's extinction. Dodos were native to the Mascarene Islands in the central Indian Ocean. They became extinct on Mauritius soon after 1680 and on Reunion about 1750. They remained on Rodriguez until 1800.
Where are viruses found?
Viruses lie dormant in any environment, land, soil, air, and on any material. They infect every type of cell, plant, animal, bacterial, and fungal.
Among the invertebrates, which are the most intelligent?
Most specialists agree that the cephalopods, the octopi, squids, and nautilus are the most intelligent invertebrates. These animals can make associations among stimuli and have been used as models for studying learning and memory. Octopi can be trained to perform many tasks, including distinguishing between objects and opening jars to obtain food.
Which important pharmacological compound was once obtained from the gymnosperm ephedra?
In the past, the drug ephedrine, used in the treatment of respiratory problems, was extracted from species of the genus Ephedra, common name, mahuang, found in China. This process has now been largely replaced with the preparation of synthetic ephedrins. Ephedra was a dietary supplement used to aid weight loss, enhance sports performance, and increase energy. In late 2003, the U.S. Food and Drug Administration banned the use of ephedra as a dietary supplement because it can pose an unreasonable health risk.
What is the controversy surrounding the midwife toad?
During the 1920s, an Austrian biologist, Paul Kammerer, 1880 to 1926, rose to prominence with claims that he had been able to demonstrate Lamarckian evolution, that is the inheritance of acquired characteristics. Kammerer had bred midwife toads, a land-breeding species, in water-filled aquaria for generations and reported that the toads had acquired the same structures that water-breeding toads use to hold onto each other. When Kammerer's toads were examined, they were found to have been injected with dye to mimic the pads found on the water-breeding toad species. Although Kammerer insisted that he was innocent, his reputation was ruined, and he committed suicide shortly thereafter.
Can all anabolic steroid use be detected?
One of the newest designer steroids is tetrahydrogestrinone, THG. This chemical is not detected by routine urine tests and has been classified as a nutritional supplement. However, according to the Food and Drug Administration, FDA, THG is related to two other synthetic anabolic steroids, gestrinone and trenbolone. Tetrahydrogestrinone is designed to boost muscle mass and strength, however. Dangerous side effects include benign and malignant liver tumors, hepatitis, aggressive mood swings, decreased fertility, acne, and an increased risk of heart attacks. A new test has been developed that can detect THG in urine samples.
What is the effect of ozone on leaves?
Ozone can enter the leaves of a plant through the stomata along with carbon dioxide, CO2. Ozone causes the cells surrounding the stomata to decrease in turgidity. thus reducing the size of the opening. This protects the plant from further exposure to ozone. However, once inside the leaf, ozone is highly reactive and can destroy the leaf cells.
What is Bayesian mimicry?
In 1861, Henry Walter Bates, 1825 to 1892, a British naturalist, proposed that a non-toxic species can evolve, especially in color and color pattern, to look or act like a toxic or unpalatable species in order to avoid being eaten by a predator. The classic example is the viceroy butterfly, which resembles the unpalatable monarch butterfly. This is called Bayesian mimicry. Bates is also well known as a colleague of Alfred Russel Wallace. In fact, it was Bates who introduced Wallace to botany and field collecting of animals and plants.
What is Berkey's Manual of Determinative Bacteriology?
Berkey's Manual of Determinative Bacteriology is an extensive reference manual used for bacterial classification. The first edition was published in 1923 under the sponsorship of the Society of American Bacteriologists. Organized in 1899 and now known as the American Society for Microbiology. This reference work was first conceived by David H. Berkey, 1860 to 1937, with the assistance of a special committee of the Society of American Bacteriologists chaired by Francis C. Harrison. The most recent edition was published in 4 volumes in 2001 under the name Berkey's Manual of Systematic Bacteriology.
What is microevolution?
Microevolution is the change in allelic frequencies that occurs at the level of the population or species. When individuals with certain traits are more successful at reproduction, the ensuing generation will have more copies of that trait. Should the trend continue, eventually the traits will become so common in the population that the population profile will change. This is microevolution.
Who introduced electrophoresis as an analytical technique?
Electrophoresis was introduced in 1937 by Arndt Salius, 1902 to 1971. A Swedish biochemist. His work pioneered the use of electrophoretic methods to separate chemically similar proteins of blood serum.
What is the difference between DNA and RNA?
DNA and RNA are both nucleic acids formed from a repetition of simple building blocks called nucleotides. A nucleotide consists of a phosphate, PO4, sugar, and a nitrogen base, of which there are 5 types. Adenine, A, thymine, T, guanine, G, cytosine, C, and uracil, U. In a DNA molecule, this basic unit is repeated in a double helix. Structure made from 2 chains of nucleotides linked between the bases. The links are either between A and T or between G and C. The structure of the bases does not allow other kinds of links. RNA is also a nucleic acid, but it consists of a single chain. And the sugar is ribose rather than deoxyribose. The bases are the same except that the thymine, T, in DNA is replaced by another base called uracil, U, which also links to adenine, A.
What is imprinting?
Imprinting occurs when an animal learns to make a response to a particular animal or object. Usually the behavior is learned by a young animal through exposure to a stimulus early in its development. There are 2 types of imprinting: filial, social attachment, and sexual. Perhaps the most famous example of imprinting is that of Konrad Lorenz, 1903 to 1989. And the Goslings who imprinted him as their mother and followed him in a classic example of filial imprinting.
Do termites have any natural predators?
Birds, ants, spiders, lizards, and dragonflies have been seen preying on young winged termites when they emerge and fly from a home colony to establish new colonies. Termites are generally most vulnerable to predators when they emerge from their home colony. Chimpanzees are also known to use sticks as tools to forage for termites.
How is nuclear waste stored and regulated?
Nuclear wastes consist either of fission products formed from atom splitting of uranium, cesium, strontium, or krypton, or from transuranic elements formed when uranium atoms absorb free neutrons. Wastes from transuranic elements are less radioactive than fission products. However, these elements remain radioactive far longer than fission products. Transuranic wastes include irradiated fuel, spent fuel, in the form of 12 feet 4 meters long rods. High-level radioactive waste in the form of liquid or sludge, and low-level waste. Non-transuranic or legally high-level, in the form of reactor hardware, piping, toxic resins, water from fuel pools, and other items that have become contaminated with radioactivity. Currently, most spent nuclear fuel in the United States is safely stored in specially designed pools at individual reactor sites around the country. If pool capacity is reached, licensees may move toward use of above-ground dry storage casks. The 3 low-level radioactive waste disposal sites are Barnwell, South Carolina, Hanford, Washington, and EnviroCare, Utah. Each site accepts low-level radioactive waste from specific regions of the country. But only EnviroCare uses above-ground storage. Most high-level nuclear waste has been stored in double-walled stainless steel tanks surrounded by 3 feet 1 meter of concrete. The current best storage method, developed by the French in 1978, is to incorporate the waste into a special molten glass mixture. Then enclose it in a steel container and bury it in a special pit. The Nuclear Waste Policy Act of 1982, as amended in 1987, specified that high-level radioactive waste would be disposed of underground in a deep geologic repository. Yucca Mountain, Nevada, was chosen as the single site to be developed for disposal of high-level radioactive waste. On July 23, 2002, President George W. Bush signed House Joint Resolution 87, allowing the Department of Energy to establish a repository in Yucca Mountain to safely store nuclear waste. However, some scientists still expressed concerns about the estimates of how long it would take for rainwater and snow to infiltrate the mountain and corrode the containers.
Is DNA always double-stranded?
At temperatures greater than 176 degrees Fahrenheit 80 degrees Celsius, eukaryotic DNA will become single-stranded. Single-stranded eukaryotic DNA does not always have the characteristic secondary helical structure. Single-stranded DNA can form a hairpin, stem, or a cross. Certain viruses have only single-stranded DNA.
How large is the arthropod population?
Zoologists estimate that the arthropod population of the world, including crustaceans, spiders, and insects, numbers about a billion million, 1018, individuals. More than 1 million arthropod species have been described, with insects making up the vast majority of them. In fact, 2 out of every 3 organisms known on Earth are arthropods. And the phylum is represented in nearly all habitats of the biosphere. About 90% of all arthropods are insects, and about half of the named species of insects are beetles.
Can pearls be genetically engineered?
Pearls can now be genetically engineered because scientists have isolated and characterized the protein nacrane, which is the primary component of pearls. When nacrane is combined with calcium ions, it forms an organic matrix similar to that found in an oyster's inner shell.
What wild flower was used by Native Americans to make red dye?
Native Americans painted their faces and dyed their clothes red with the root of the bloodroot wildflower. Sanguinaria canadensis, which is also called redroot, Indian paint, and tetawort. Bloodroot, found in shady, damp, and woodsy soils. Blooms in May and has white flowers that are 2 in 5 centimeters wide.
What was the first commercial use of genetic engineering?
Commercial recombinant DNA technology was first used to produce human insulin in bacteria. In 1982, genetically engineered insulin was approved by the FDA for use by diabetics. Insulin is normally produced by the pancreas. And for more than 50 years the pancreas of slaughtered animals such as swine or sheep was used as an insulin source. To provide a reliable source of human insulin, researchers harvested the insulin gene from cellular DNA. Researchers made a copy of DNA carrying this insulin gene and spliced it into a bacterium. When the bacterium was cultured, the microbe split from one cell into 2 cells. And both cells got a copy of the insulin gene. Those 2 microbes grew, then divided into 4. Those 4 into 8, the 8 into 16, and so forth. With each cell division, the 2 new cells each had a copy of the gene for human insulin. And because the cells had a copy of the genetic recipe card for insulin, they could make the insulin protein. Using genetic engineering to produce insulin was both cheaper and safer for patients. As some patients were allergic to insulin from other animals.
What is the difference between an African elephant and an Indian elephant?
The African elephant, Loxodonta africana, is the largest living land animal. Weighing up to 8.25 tons, 7,500 kilograms, and standing 10 to 13 feet, 3 to 4 m, at the shoulder. The Indian elephant, Elephas maximus, weighs about 6 tons, 5,500 kilograms, with a shoulder height of 10 feet 3 meters.
What is biogeography?
Biogeography is the study of the distribution, both current and past, of individual species in specific environments. One of the first biogeographers was Carolus Linnaeus, 1707 to 1778. A Swedish botanist who studied the distribution of plants. Biogeography specifically addresses the questions of evolution, extinction, and dispersal of organisms in specific ecosystems.
What is the carbon cycle?
To survive, every organism must have access to carbon atoms. Carbon makes up about 49% of the dry weight of organisms. The carbon cycle includes movement of carbon from the gaseous phase. Carbon dioxide CO2 in the atmosphere, to solid phase. Carbon-containing compounds in living organisms, and then back to the atmosphere via decomposers. The atmosphere is the largest reservoir of carbon, containing 32% CO2. Biological processes on land shuttle carbon between atmospheric and terrestrial compartments. With photosynthesis removing CO2 from the atmosphere and cell respiration returning CO2 to the atmosphere.
What is taxol?
Taxol is a drug used to treat ovarian cancer. It freezes cancer cells early in the process of cell division. When the cells are unable to divide, they eventually die. Taxol is obtained from the bark of the Pacific yew, Taxus brevifolia. A gymnosperm that grows in the Pacific Northwest. Because the Pacific yew is a small tree that grows slowly and is not found in abundance, researchers have synthesized taxol.
Can animals hear different sound frequencies than humans?
The frequency of a sound is the pitch. Frequency is expressed in hertz, hertz. Sounds are classified as infrasounds, below the human range of hearing, sonic range. Within the range of human hearing, and ultrasound, above the range of human hearing.
What is a transposon?
A transposon is a nucleotide sequence in a gene that literally jumps around from one chromosome to another. Transposons can be problematic, as they may disrupt the normal function of an important gene by their random insertion into the middle of that gene. However, transposons are found in almost all organisms, and their presence in a genome indicates that genetic information is not fixed within the genome. By studying results of the Human Genome Project, scientists predict that about 45% of all human genes may be derived from transposons.
How did Hugo de Vries demonstrate sympatric speciation?
Hugo de Vries, 1848 to 1935, discovered a way in which a population could become a separate species while still sharing the same environment with other members of the species. The process, known as sympatric speciation, occurs almost exclusively in plants rather than in animals and involves a series of rare genetic accidents that can occur during the formation of gametes, eggs and sperm. As a result, Gametes are formed as polyploids, that is, they have extra copies of each chromosome and thus are unable to match their chromosomes to others of the same species. Since these polyploids are forced to mate only with other polyploids in the population, they are reproductively isolated and considered a new species.
What is a spliceosome?
A spliceosome is a structure, comprised of RNA and protein, that is responsible for RNA splicing in the eukaryotic nucleus.
What is genetic engineering?
Genetic engineering, also popularly known as molecular cloning or gene cloning, is the artificial recombination of nucleic acid molecules in a test tube, their insertion into a virus, bacterial plasmid, or other vector system, and the subsequent incorporation of the chimeric molecules into a host organism in which they are capable of continued propagation. The construction of such molecules has also been termed gene manipulation because it usually involves the production of novel genetic combinations by biochemical means. Genetic engineering techniques include cell fusion and the use of recombinant DNA or gene splicing. In cell fusion, the tough outer membranes of sperm and egg cells are removed by enzymes, and then the fragile cells are mixed and combined with the aid of chemicals or viruses. The result may be the creation of a new life form from two species, a chimera. Recombinant DNA techniques transfer a specific genetic activity from one organism to the next through the use of bacterial plasmids. Small circular pieces of DNA lying outside the main bacterial chromosome, and enzymes, such as restriction endonucleases, which cut the DNA strands, reverse transcriptase, which makes a DNA strand from an RNA strand, DNA ligase, which joins DNA strands together, and TAC polymerase, which can make a double-strand DNA molecule from a single-strand primer molecule. The recombinant DNA process begins with the isolation and fragmentation of suitable DNA strands. After these fragments are combined with vectors, they are carried into bacterial cells, where the DNA fragments are spliced onto plasmid DNA that has been opened up. These hybrid plasmids are then mixed with host cells to form transformed cells. Since only some of the transformed cells will exhibit the desired characteristic or gene activity, the transformed cells are separated and grown individually in cultures. This methodology has been successful in producing large quantities of hormones, such as insulin, for the biotechnology industry. However, it is more difficult to transform animal and plant cells. Yet the technique exists to make plants resistant to diseases and to make animals grow larger. Because genetic engineering interferes with the processes of heredity and can alter the genetic structure of our own species, there is much concern over the ethical ramifications of such power. as well as the possible health and ecological consequences of the creation of these. Bacterial forms. Some applications of genetic engineering in various fields are: agriculture, crops having larger yields, disease and drought resistancy; bacterial sprays to prevent crop damage from freezing temperatures, and livestock improvement through changes in animal traits. Industry, use of bacteria to convert old newspaper and wood chips into sugar. Oil and toxin-absorbing bacteria for oil spill or toxic waste cleanups, and yeasts to accelerate wine fermentation. Medicine, alteration of human genes to eliminate disease, experimental stage. Faster and more economical production of vital human substances to alleviate deficiency and disease symptoms. But not to cure them. Substances include insulin, interferon. Cancer therapy, vitamins, human growth hormone ADA, antibodies, vaccines, and antibiotics. Research, modification of gene structure in medical research, especially cancer research. Food processing, rennin, enzyme, in cheese aging.
How are fibers classified?
Fibers are classified in several ways. One way to classify fibers is based on their location, that is, whether or not they are in the xylem. Fibers in xylem are called xylary fibers, while those found in other tissues are called extraxillary fibers. Extraxillary fibers are usually longer than xylary ones. Fibers may also be classified according to their hardness. Hard fibers are from monocots and include xylem, thus making them lignified and stiff. An example of a hard fiber is rope made from sisal, Agave cisalina. Soft fibers, also called bast fibers, are harvested from dicots lacking lignin and are generally stronger and more durable than fibers from monocots. An example of a soft fiber is linen from flax, Linum usitatissimum.
What are the steps of microscopic autoradiography?
In microscopic autoradiography, the desired radioactively labeled compound is first added to cells or organisms, and the material is then incubated. After a period of time to allow the radioactive compound to become incorporated into newly forming intracellular molecules and structures, the incubation is stopped, and the biological specimen is rinsed to wash away the excess radioactive compound. The specimen is prepared in a conventional manner, sectioned and placed on a microscopic slide. The slide is then covered with a thin layer of photographic emulsion. Next, the prepared slide is placed in a sealed box for an appropriate period of time, several days to several weeks, to allow the radioactivity in the cell to expose the emulsion above it. Upon removal from the sealed box, the emulsion is developed, and the specimen is examined under the microscope.
What are the main components of a bacterial cell?
The major components of a bacterial cell are the plasma membrane, cell wall, and a nuclear region containing a single, circular DNA molecule. Plasmids, small circular pieces of DNA that exist independently of the bacterial chromosome, are also present in a bacterial cell. In addition, some bacteria may have flagella, which aids in movement, pili or fimbriae, which are short, hair-like appendages that help bacteria adhere to various surfaces, including the cells that they infect. Or a capsule of slime around 92, the cell wall that protects it from other microorganisms.
What is a seed?
A seed is a mature, fertilized ovule. It consists of the seed embryo and the nutrient-rich tissue called the endosperm. The embryo consists of a miniature root and shoot. Once the seed is protected and enclosed in a seed coat, it ceases further development and becomes dormant.
How widespread is the use of GMO crops?
As of 2002, more than 120 million acres, 40 million hectares, of fertile farmland were planted with GMO crops. The acreage is confined to 4 countries: United States, containing 68% of the total acreage; Argentina, 22%, Canada, 6%, and China, 3%.
Why is liquid water more dense than ice?
Pure, liquid water is most dense at 39.2 degrees Fahrenheit, 3.98 degrees Celsius, and decreases in density as it freezes. The water molecules in ice are held in a relatively rigid geometric pattern by their hydrogen bonds, producing an open, porous structure. Liquid water has fewer bonds, therefore, more molecules can occupy the same space, making liquid water more dense than ice.
Who is considered the father of lichenology?
Eric Acharius, 1757 to 1819, is considered the father of lichenology. He was the founder of modern lichen taxonomy, having described and arranged lichens into 40 distinct genera. His 4 major works of research: Lichenographum Swaix Prodromus, 1798. Methodus Lichenum, 1803, Lichenographia Universalis, 1810. And Synopsis Methodica Lichenum, 1814, formed the foundations of modern lichenology.
Who introduced electrophoresis as an analytical technique?
Electrophoresis was introduced in 1937 by Arndt Salius, 1902 to 1971. A Swedish biochemist. His work pioneered the use of electrophoretic methods to separate chemically similar proteins of blood serum.
When was the nucleus first described?
The Scottish botanist Robert Brown. 1773 to 1858, first named and described the nucleus in 1831, while studying orchids. Brown called this structure the nucleus, from the Latin nucula, meaning little nut or kernel.